Michael, I do appreciate that. Good evening, everybody. And I bring you greetings from the Dallas-Fort Worth area. And this is the last talk that I'm giving by way of the AzOSA, and Joseph is the president, El Presidente, as we say in China. And so it is good to be here. And this is my swan song. This is the last presentation in about 2 weeks. And so what we're going to look at, as Michael said, is pathways to God, metabolic pathways. All animals, everybody, all animals and people have metabolic pathways. If you had supper tonight, right now your supper is being digested by an incredibly complex and sophisticated series of metabolic pathways. So, Frank, you're saying metabolic pathways, what do you mean? Well, it's a series of enzyme catalyzed biochemical reactions in a cell in which the products of one reaction becomes the substrate for the next. Don't worry, we'll go ahead and define what those terms are. It has to do with the enzyme substrate complex. Metabolic pathways are used to convert one compound into another. Very important, okay? Because we need things like carbohydrates, fats, and proteins. And so we get those by these metabolic pathways. Now, does anybody know, besides Dr. Cazell, what these are? Or Michelle, because they both know what these are. Very good. They're cells. They're what we call simple squamous cells. And so I used to teach introductory biology classes to pre-meds and also to nursing students. And I would have them use their fingernail and just scrape off some squamous epithelial cells from the inside of their cheek. And then they put it on the slide. And ideally, that's what it looks like. And so these dark staining centers are called a what class? Very good. See how easy biology is? It's just so simple. Okay. And so what about these simple squamous cells? Well, as a matter of fact, this is what Chuck Darwin saw. He saw something very close to this. And Darwin in 1859 thought, well, a cell is just a simple structure. It's very small, and it's protoplasmic, and it has a center that stains darkly. And literally, that's all he thought. He never realized how incredibly complex the metabolic factory is inside these cells and all the organelles, the very, very tiny structures within the cell itself. Okay, well, metabolic pathways are found in all living creatures, whether it's the paramecium that you looked at under the microscope in high school biology. Wasn't that boring? And mammals and the fish and the toadstools, the Basidiomycetes, all of that has metabolic pathways, and it all comes down to this right here. It's called the enzyme substrate complex. Now, very quickly, keeping all the cookies on the bottom shelf, the enzyme is a very, very sophisticated protein molecule. And the enzyme has, it says here, an active site. This is where the biochemical reactions occur. And so when the substrate, that which is going to be broken into products, fits into the enzyme perfectly, within just a fraction of a millisecond, that's no exaggeration, just as soon as a substrate touches the enzyme into the active site, it changes immediately. Now, the enzyme doesn't change, but the substrate does. It breaks into two or more products. And it happens, as I say, within a fraction of a millisecond. Well, Frank, you kind of lost me. What do you mean by that? Okay, here's another diagram that kind of shows what's going on. This is the enzyme molecule we just previously saw. Before I get into anything else, this enzyme here and here doesn't change at all, does it? It doesn't fracture, it doesn't break, it doesn't change its shape. The enzyme stays the same through this whole metabolic process that takes a fraction of a millisecond. So this is a substrate. They also call it a reactant molecule. Here it is. This is the active site of the enzyme. It comes together in a fraction of a billionth of a second and then splits into the 1, 2 products you see there. And notice the enzyme doesn't change. Right now, this is going on in your body, hand over fist. It's very, very rapid. Again, this is a simple metabolic pathway. This is what we're talking about this evening. And again, I'll keep it very basic. But you have a starting molecule. This is a substrate, not the enzyme. The enzyme is here. The substrate fits against the enzyme, and boom, you get products. And then this product here fits into another enzyme, and it changes, and it changes. So you have 1, 2, 3, enzymatic reactions where the enzymes do not change and the products do. The starting molecules, A, B, C, and D, do change based on the enzyme. So that's basically what's going on in your GI tract, and as a matter of fact, in all areas of every one of your trillions of cells that make up your body. The product of one reaction becomes the substrate for the next B there, C and D. So that's basically it. This is another way of looking at a simple, quote unquote, there's nothing simple about metabolic pathways, but this is a basic metabolic pathway, starting with one molecule and one enzyme, resulting in a second molecule. The second enzyme works on that, a third molecule, the third enzyme works on that, and around you go. In biochemistry, a metabolic pathway is a linked series of chemical reactions that occur within the cell. Once again, this is going on very, very rapidly in your trillions of cells. This is a typical animal cell or a person cell. I keep animals and people separate. Why is that? In Genesis chapter 2, God created us in His image. He didn't create animals in His image, just us. And so we are separate from the animal community as much as the evolutionary community would wish that was not the case. And so we have a typical cell here with the cytoplasm. This is where all these enzymatic reactions occur, the DNA within the nucleus of the cell. The mitochondria, remember that from high school? The energy producer of the cell, ATP energy, the high energy bonds break and releases energy and so forth. This is due to the Krebs cycle, and that Krebs cycle is very complex, and very basically, the Krebs cycle in the mitochondria looks like this. You have a 2-carbon molecule, it hooks up with the 4-carbon molecule, which is coming up around here. 4 plus 2 is 6-carbon molecule, okay? Comes around, loses one of the carbon molecules when we breathe in oxygen. Right now, you're breathing in oxygen, and you're going into your cells, and that oxygen hooks up with that one carbon there. And you exhale what? Carbon dioxide. How important is it that the oxygen combines with those carbons and you get rid of it and exhale? How important is that? Well, just hold your breath and see how long it takes before these carbons start building up. And you, as much as you try not to breathe, you have to breathe, because the carbon builds up very, very quickly because of all these metabolic reactions and so forth. Here's a 5-carbon molecule. Here's nicotinamine adenine dinucleotide phosphate. It's coming around. Oh, we're losing another carbon here. 5 minus 1 is 4. There's a 4-carbon molecule. Here's another flavin adenine dinucleotide, which is picking up another of hydrogens, not the carbons. And so the Krebs cycle that's going on right now is taking a 2-carbon acetyl group and hooking it to a 4-carbon, 6-carbon. You breathe in some oxygen, get rid of a carbon, and goes around and around and around. Whether it goes clockwise or counterclockwise, it doesn't matter. And so, there's something else called the pyruvate dehydrogenase complex, where one carbon is carved off of a 3-carbon pyruvic acid molecule, giving you the 2-carbon acetyl group there, happening all the time. Very, very sophisticated. Yes?
When was this discovered?
This is covered in the 1930s. Yeah. And so, as a matter of fact, the discoverer, his name was Dr. Cycle. And they don't know why they call it Krebs, but it was Dr. No, I'm joking. It's called the Krebs cycle because it's a cyclic process. And so he got the Nobel Prize in Medicine for discovering that. Yes, sir?
You derive energy from this process.
Yes, you derive energy. Where's the energy from? Right here. The nicotinamine adenine dinucleotide phosphate picks up these high energy hydrogens, these protons, and it carries it. And then the protons, it's oxidative phosphorylation, where adenosine diphosphate gets phosphorylated to adenosine triphosphate, and then you get all this energy. So it gets pretty complex. But here it is, cutting to the chase. In each moment of cell life, billions of substrates, and this is an evolutionist speaking, billions of substrates are transferred into billions of products by billions of enzyme molecules. Sounds like Carl Sagan, doesn't it? Okay, these reactions are extremely fast. This is putting it mildly. These reactions are occurring with a fraction of a billionth of a second. And we can imagine the cell as a viscous environment where these reactions occur in an ordered and only apparently chaotic. I like that. It's only apparently chaotic, but it's finely tuned. It's symphonic. It's what God has designed in each one of our cells. We give glory to the Creator and not the creation, as the Apostle Paul says in Romans 1. And only apparently chaotic fashion. The whole body of these reactions is called metabolism. So now, in just these few minutes here in church, you get a new appreciation when you hear that complex word, metabolism. Just think of that Krebs cycle. Just think of all of these processes that are going on. And so this guy is no creationist, and I appreciate that he's explaining this very basically, fundamentally. Here's a question of the evening right here, folks. Chance or design? These metabolic pathways that I just described to you, and that goes into a lot more detail, did it come about by blind random chance, or did it come about by plan and purpose and special creation? Those are the two choices. Unfortunately, colleges and universities are saying dumb, lucky, blind, random chance. That's how you get these metabolic pathways. And if I could just, I don't want to be too intemperate this evening, but that's crazy. That's just absolutely crazy. There's no way something like that can happen. Nota Garcia and other authors address the still unsolved question of how metabolic pathways have evolved. Did you get that? Yes. When was this said? This was said way back in 2018. That's just a few years ago. Let's read that again. The authors address the still unsolved question of how metabolic pathways have evolved. Could I raise my hand to the back of the classroom, maybe hold a handkerchief and wave it a little bit and say, you know what? It never did evolve. And they're going to keep searching for how it evolved, and they'll never come to a knowledge of the truth because they refuse to give glory to the Creator, and instead they say, how did time and chance and natural processes ever produce this? And they will never come to an answer. It simply is beyond this idea of time and chance. From the last universal common ancestor, it's called the LUCA, last universal common ancestor, to present times. And so this is the evolutionary tree of life. Whenever you see something like this, class, remember this. This is religious. This is philosophical. This has no bearing in good, observable, repeatable science. And so this idea of a last universal common ancestor giving rise to the archaea and the eukarya and the bacteria, no. It never happened. Remember what science means. Science means, the main thing it means is observation. Science means observation. Nobody ever observed this occurring. This is nothing more than a guess. The emergence of the pathways, and that's what we're talking about this evening, that now comprise core and central metabolism, there's that word, or even intermediate metabolism, is particularly puzzling. Well, yes, they're getting it. Of course it's particularly puzzling. They'll never come to knowledge of the truth. Again, this is in the year 2018, just a few years ago. And so this is here, there it is. There's the tricarboxylic acid cycle, or the Krebs cycle. It goes by a variety of names. Fatty acid cycle, that's very, very important too. There's so much you say. This alpha ketoglutarate acid right here, just this one molecule, my graduate professor said, Sherwin, before you get your master's, you should be able to write 12 pages front and back on just this one molecule right here, just this one molecule, 12 pages front and back. So I started practicing, but fortunately he never asked me that. There are three theories regarding the evolution of metabolic pathways, okay? So the evolutionists are not going to say yes to creation, because they're evolutionists. They don't read the Bible, they could care less. Hey, I'm an evolutionist, I'm a smart researcher, I don't need religion. But that puts them in a very difficult position of trying to explain something like this. So what are the three theories regarding the evolution, so-called, of metabolic pathways? First one is called the semi-enzymatic theory. The second theory is the retrograde hypothesis. And the third one is the patchwork hypothesis, okay? So these are the three theories that evolutionists have come up with to try and do some kind of damage control and say, yes, this is how these metabolic pathways that are so insanely complex evolved by time and chance. And so I did a little research. First of all, I found out these are the three theories that they use. And so at ICR, I did some research. And I have these three books in my office. I've got lots of books in my office, and I even read one or two of them on occasion. But anyway, this is a third edition of a book called Evolution. The guy's name is Douglas Fujiyama. He hates creationists. That's the best I can put it. He will go on film, he'll go on video to vent his spleen against creation science. So he's diametrically opposed to what I'm saying right here tonight. So Fujiyama, he's about my age, and he has a third edition of Evolution. Then we have the Principles of Biochemistry, and Leninger. I used Leninger when I was in graduate school. He's since gone on to meet his creator, but other researchers have continued Leninger's textbook, and that was 2017, fairly recently. And then Molecular Cell Biology by 8 authors. Okay, so you have these 8 authors, you have these authors here, you've got Fujiyama, and do they know of these 3 theories? Are these 3 theories mentioned in these 3 textbooks? As we say in Mexico, nada. Okay, none of these 3 theories. I checked the appendix. I checked the chapter headings. I did all that I could besides just reading every last word of the book there to see if those 3 theories are mentioned. They are simply not mentioned in the books where they should be mentioned. Nada, nothing, niet. So this is Escherichia coli bacterium, E. coli. Maybe some of you have heard of E. coli breakouts, where people get poisoned by some subspecies of Escherichia coli. And look at all those little projections coming off of those. Those are called flagella. And some subspecies of the genus species E. coli, some subspecies have the flagella coming off there. And it looks like, you know, whip-like appendages, which is exactly what they are. And so the creator has designed this bacterial cell. It's not like our cells are built. And the flagella actually rotates, it spins. And so researchers were intrigued by this, and they decided to fracture, that is, bust up these cells and see if they could expose the engine, the motor that makes the flagella turn. So we're going to look right now very quickly at how this flagella goes into the side of the bacterial cell. Now they're going to break it open, expose the motor, expose the engine, and this is what they found. Okay, now this is a diagram, this is a drawing, this is an illustration, but look at that white and black grainy picture there. You know what that is? That is actual picture using an electron microscope. Most of us have heard of an electron microscope, right? Things that are magnified thousands of times. And so using an electron microscope, they found a portion of, yes, like literally finding a needle in a haystack. But they did find the motor, and they used an electron microscope to highly magnify it, and that's what they got. And so they got an illustrator who's no creationist. He doesn't have any religious axes to grind or anything. And so the illustrator drew this. It's about here, ladies and gentlemen, the hair in the back of my neck went up. Because when you start looking at this, especially if they start using words like motor and rod and the filament and all that, they're using engineering words. And as time went on, they began to study this even more, and even more complexity of this flagellar motor appeared. So this is an axle, right? You go outside in one of these cars here, look at the rear axle, it looks something like this. Everybody on this planet, even evolutionists, would say that axle is designed. Now, my youngest is an engineer at Fiat Chrysler in Detroit. He got his degree in what? Engineering, okay, in mechanical engineering. And so he's in graduate school now, but he's an engineer, and he assures me that these have to be exquisitely designed and then manufactured to very precise measurements. Well, the evolutionists would say yes to this, right? Who wouldn't? Who would deny that this isn't designed, okay? But they'll look at the flagellar motor that's infinitely more tiny, almost 1,000,000 times smaller than this, and they say, nope, that's not designed. Not designed? Are you kidding me? Let's take a closer look. Both of these have a drive shaft. Both of these have a bushing. They have a universal joint. That motor that they say isn't designed has a universal joint. It has a rotor, rings, and a clutch. Stop right there. A clutch. Are you kidding me? This thing has a clutch. Yep, National Science Foundation found out about 10 years ago as they did more research that the flagellar motor has a clutch, and here it is. Here's where the clutch is engaged. This is what the gear looks like. And this is when the molecular clutch is engaged, and then when it's disengaged, it's see there. So this flagellar motor can be engaged and disengaged depending on the situation. Depending on the situation, what do you mean? This is just a bacterial cell. It doesn't think, it doesn't know what to do. Well, yes and no. So they began to do some research. And this is the motor system of that little tiny E. coli that you can barely see with a light microscope. My students who were pre-med and pre-nursing had fits trying to see individual E. coli bacteria under the microscope. And they'd, Mr. Sherwin, could you, and then we'd have to focus in and find it. It's just very, very tiny. And so this is the motor system of that tiny bacterial E. coli cell. And you can see nothing is left to chance as this motor system is built. The bacteria can tumble, it can swim, it can respond to various stimuli, it can have counterclockwise and clockwise rotation based on a switch, which is also based on the rotary mechanism. It is very, very sophisticated, very quick. But see, that makes no sense unless the bacteria knows where to go. And what do you need to have to know where to go? You have to have a sensory system. So researchers found out that the E. coli bacterium, which is infinitely tiny, has a sensory system. It actually can sense where to go and where not to go. So if some area of the aquatic environment has a high hydrogen ion concentration, that means it's very acidic. And so the tiny bacterial cell knows, quote unquote, not to go there and to turn around and go the other direction. It's just a bacterial cell. Also, there might be some food, some fatty acids or whatever else, and so this little tiny bacterial cell will know, quote unquote, to go to that area where there's food and to avoid areas that have a high acidic concentration. Look at this. All of this. Here's the ATP that helps with the methylation, the gradient inference, the aspartate signals, which is an amino acid that helps in signaling. All sorts. Well, let's superimpose one on the other because it's going on at the same time, right? They're both going on in the same time inside of this tiny bacterial cell. But you remember the energy acquisition where a cell needs energy, ATP? Well, adenosine diphosphate has to be phosphorylated into adenosine triphosphate, and the way that happens is nicotinamine adenine dinucleotide phosphate carves off these hydrogen ions and carries them to a special system which helps to phosphorylate these adenosine diphosphates. It gets very, very complex. Oops, I forgot to tell you. Inside of this bacterial cell, at the same time, the DNA molecule, which is a highly circuitous molecule. Maybe some of us are old enough to remember those old-time phone cords, which were, and they always got, you got mad because they would always, what do they call those phone cords? The coiled phone cords or whatever. Well, the DNA, deoxyribonucleic acid molecule of the E. coli is supercoiled. And it's packed up, supercoiled inside where all of this is going on at the same time. And when the E. coli has to reproduce itself, that DNA molecule, that circular chromosome molecule, has to be reproduced with exquisite fidelity. And you have two sister cells from that. All of that is going on at the same time. And what do secular professors say to students who say, you know what, I really think this might be designed? You know what the secular evolutionary atheist professors tell the students who question this idea that it could come about just by time and chance? They say something like this: how can you seriously believe that all of this is a result of intelligence? That's a facepalm. That's a double facepalm. What did the Apostle Paul say in Romans chapter 1 and verse 20? He said that God's creation is not only seen, but Paul said God's creation is clearly seen. And I think that's what Paul is talking about here. Paul didn't know anything about pyruvate dehydrogenase. He didn't know anything about what's going on within E. coli. He didn't even know what bacteria was. But he knew that God's creation is not only seen, but it's clearly seen. Paul could see the sunset. Paul could look at his hand and say, wow, look at that. How did that form? God's creation is clearly seen. And if you're still kind of on the fence as to whether something like this can come about by time and chance, every graduate school in the nation, I know at the University of Northern Colorado, we have this poster as well. And this is a poster of what's going on inside just one of your trillions and trillions of cells. Okay, there's the acetyl-CoA before it goes into the Krebs cycle there. Pyruvate. Pyruvate is a 3-carbon energy-rich compound. Here it is. It's entering into the mitochondria. It is undergoing what's called glycolysis. A glycolysis is 11 steps, 12 specific enzymes that we talked about just a few minutes ago. That's just glycolysis. That's just taking glucose in your blood and going into the cell, and by glycolysis breaking that 6-carbon molecule into 2 energy-rich pyruvate molecules that then enter into. But all of that is also going on as well. All of the amino acids that help to make up protein molecules, all of the fatty acids, all of this is going on. And remember, what our evolutionary professor says to the students who question that this could come about by chance, he says, how can you seriously believe that this is designed? And you just have to laugh, because this is Romans 1:20 right here. God's creation is clearly seen. And you know what this reminds me of? When I look at this, I'm going, you know, I've seen something like this before. There's a store that's no longer around, but I used to keep them in business for 20 years, and that store is called Radio Shack. And Radio Shack's not around anymore, but Radio Shack was very busy with this, right? Where you get all these printed circuits and all. Don't these printed circuits remind you of what's going on in our cell? It really does to me. And everybody knows that you get these highly sophisticated and complex printed circuits by explosions in Radio Shack. Okay. That's how you get that. Well, my field is parasites, okay? I'm an invertebrate zoologist. I study parasites. And by the way, you don't have to go to France to study parasites. Thank you. Thank you. I'll be here all evening. Okay. Yeah, there we go. See, that's coming at you, coming at you. And so my field is parasites, and so this is a textbook that my late professor used. I was his graduate student in the early 1980s. He was a dyed-in-the-wool atheist. He told me before I signed up to be his graduate student, he sat back in his creaky office chair on the 6th floor of Ross Hall of Science in Greeley, Colorado, and he said, you have to understand, I'm an atheist, and I require all of my students to take a course in evolution. I said, no problem. Because by that time, I knew there was no facts of evolution. And whatever the professor was going to tell me in that evolution class, it wasn't fact after fact after fact of evolution, because such facts simply do not exist. And so his name was Dr. Gerald Schmidt. And Gerald Schmidt literally wrote the book on parasites. And I was very much honored to be his grad student, even though we were like this. Here I am, here he was. And really, a graduate student and the professor should be like this. Should, you know, as the professor just pours his life into his graduate student. That's the way it should be ideally, but it wasn't. That's okay. I got my master's degree, but this is a book, Foundations of Parasitology, and you can't see the name there, but it's Gerald Schmidt. And he's the one that literally wrote the book on parasites. Now, why am I making a big deal about this? Because on page 20 of Foundations of Parasitology, this is long after Schmidt has died, they said, a multitude of other metabolic events that make biochemical pathways, that's our subject tonight, look like printed circuits of high-tech electronic equipment. Thank you so much. I bow before these professors that gave the analogy of these high-tech printed circuits that took a lot of sophisticated detail and some brilliant minds and the best research available to make those printed circuits. And so they're giving the analogy that that's what the biochemical pathways. Look, if it takes somebody brilliant and millions of dollars to make those kind of printed circuits, shouldn't that be the same for the biochemical reactions, which are infinitely more complex? Just like our example of the axle of a truck and the rotary flagellum of that bacterium, which is more complex. That bacterium, hands down, that bacterium is more complex. So this is evidence of design. Design means a designer. Creation means a creator, okay? If things look intelligently designed, there just might be an intelligent designer. So regardless, evolutionists have no choice. They cannot say, boy, well, this really looks like maybe God created this. Boy, an evolution, what would they stop being? An evolutionist, okay? They said that if God did it. So evolutionists have no choice but to say these complex pathways evolve through time and chance and something they call pressure. Pressure, okay, I know a little bit about biology, a little bit, and I have no idea what that word means in the biological sciences, but they use it. Yeah, that's it. Yes. Bing, bing, bing. Pressure to conform. I'm going to use that. I'm glad I thought of that. I'm going to use that. Okay. And here's an evolutionary explanation. The emergence and evolution of metabolic pathways, that's what we're talking about tonight, represented a crucial step in molecular and cell evolution. In fact, the exhaustion of the prebiotic supply of amino acids and other compounds that were, there's a good scientific word there, what is that? Likely present on the primordial earth imposed an important selective, there's that word again, selective pressure. What's a selective pressure? You know, can I look at it? Can I break it? I mean, you know, what is it? Favoring those primordial heterotrophic cells. And that was said in 2012. But, you know, selective pressure, when I see that phrase or I hear somebody use that, a little warning bell goes off because I think I'm being taken for a ride, okay? Because I don't know what a selective pressure is. Here it is from Leninger's biochemistry text. This is a standard biochem text in the United States, if not the world. And so I use this in graduate school. Yeah, that's it. Other what? Pressures must also have operated throughout evolution, selecting for, well, there's a religious phrase, selecting for, how do you select for something? If it's all random, if there's no creator, how do you select for it? Regulatory mechanisms. And this is Leninger's text in just a few years ago. Evolution states the emergence and evolution of metabolic pathways represent a crucial step in the molecular and cell evolution. In fact, the exhaustion of the prebiotic supply and all that we just read there, and selective pressure is what they're talking about. So here are the evolutionary wiggle worm words that they use. Emergence of and evolution, likely, selective pressure, favoring, which became, okay? None of that is scientific. All of this is very philosophical. Let's see. Okay, here's another one from a different evolutionist in 1999. A likely scenario, well, that's not science. Likely scenario of evolution of biosynthetic pathways is believed. Boy, well, I love that word. That's a great word for the Christian, believed, okay? To have occurred by retroevolution. What does retro mean? Going backwards. So evolution is amazing. It can do anything. It can go forwards, it can go backwards. Retroevolution through recruitment of existing enzymes rather than generation of de novo classes. That is, the production of new classes. It's been proposed that such retroevolution occurred in steps as a response to depletion of essential metabolite and availability of other related substances in the environment. In this article, I argue that because of instability of many such extant, that is present right here and now, extant intermediates, it's unlikely that retroevolution had occurred in steps. So he's appealing to retroevolution, but he's adding a lot of wiggle worm to it, wiggle words. And so this is not science. That's the point I'm trying to get across. This is gobbledygook, but it's not solid, empirical, investigative research. Such evolution in many cases has taken place by jumps. Okay, so they, regardless, will say it occurred by jumps, specifically by recruitment of a multifunctional enzyme. Stop right there. Enzymes are complex. Okay, you have that critical active site there, but to have a multifunctional enzyme, you have just increased the complexity by at least a factor of 10. And so they're just suggesting that multifunctional enzyme capable of catalyzing several steps at a time, albeit inefficiently, okay? So they're saying, oh, it wasn't perfect at that time, it was inefficient. I'm going, again, this is not a scientific explanation. It's like saying, well, no fireman is going to fall all the way up to the top of a building. That's ridiculous. And so firemen can fall by steps to the top of a building. Well, that's also preposterous. That people don't fall up, whether it's in one jump or in just a series. The pathway may have evolved by a single leap. Gene duplications and further evolution to more efficient enzymes led to extant, that is, present pathways. Such a mechanism predicts that some or all enzymes of a pathway must have, there's the religious statement, descended from a common ancestor. By the way, have we ever found that common ancestor? Never found that common. Will we ever find that common ancestor? No. According to the Bible, we never will find that common ancestor. Remember what it says in Genesis chapter 1? God created after their kind. How many times do we see that phrase in Genesis 1? 10 times. 10 times God told us in one chapter that he created after their kind. Okay, so that means that God did not use evolution. He created after their kind. He didn't use evolution. And so here's the same situation here. Descended from a common ancestor, we'll never find that common ancestor. It never existed. We're running out of time here. This is what, when I joined ICR 1996, I was able to go to the University of UCSD, University of California, San Diego, and take night classes in some of the biochemistry classes, including G protein coupled receptors. So Frank, what is that? Well, it's part of this metabolic pathway, but it is extremely complex. And so I took 2 semesters of this G protein receptors. It's called molecular pharmacology. That was the name of the class. Make up a large family of membrane receptor proteins. This is the membrane receptor protein. This is the first messenger. It's called a ligand. It fits in there, just like we saw with the enzyme. Yeah, very good, the enzymes. And it causes a change. It rings a bell in the cytoplasm of the cell within a fraction of a second. That detects molecules outside the cell and activates internal signal transduction pathways. There they are. And ultimately, cellular responses down here in the DNA of the nucleus. So we had 2 semesters just to appreciate this and go into a lot of detail of what's going on here. And I came away with my head swimming, saying, how can time and chance produce something like that? We have been studying the very thoughts of the Creator. And that's the thing about being a creation scientist, especially in 21st century. We want to emphasize that doing this kind of research and appreciating this kind of complexity is a form of worship. It's a form of worship, it truly is. As we give glory to the Creator, but not the creation. And that's what Paul warns about in Romans chapter 1, doesn't he? He warns in the last days of which we're living in, that man will worship the creation and not the Creator. Well, this is another G protein coupled receptor, and Joseph and I were just talking about this today, I think. The second messenger, protein kinase A, and protein kinase A that's been activated by cyclic AMP. Very, very sophisticated process occurring. It's called intracellular signaling pathway, and it's going on right now as you're sitting here in your trillions of cells. Here's Leninger again, signaling mechanisms. Here's the religious statement. Must have. Okay, that's a religious statement. Well, it must have. You imagine if the evolutionist heard a creationist say, well, it must have been that way as a creationist. They wouldn't let us forget that. So why did the evolutionists get away with it? And by the way, they even print it in their book. Must have is not a scientific explanation. The sense of smell, you know, whether it's Chanel number 5 or a pizza with anchovies. I love anchovy pizzas. and whatever you're smelling there, this is what's going on. It's these G protein coupled receptors in your nasal epithelium, and that helps you to appreciate the varieties of smells and odors and all that. And look at this, it was in the 21st century, 2014, little is known about how olfactory receptors function in mammals, and we would also add people, okay, or how this large gene family has evolved. They don't know as of the 21st century. Thank you for admitting it. And who said that? National Science Foundation, okay? And in response to different evolutionary challenges. So they hold on tenaciously to evolution theory, even though it's not giving them any explanations whatsoever. We can see the fingerprint of the Creator right here. And you know, there's some smells that you haven't smelled in decades, and you get that smell again. For me, yeah, I live in the Chicagoland area, it's hot tar. I smell hot tar, it takes me back to my childhood. Boom, just like that. And so it's amazing what's going on in our brain when we have that type of thing occurring. Well, I'm not going to get into gene duplication because this really does get complex. But basically, gene duplication is the most widely known mechanism responsible for the origin, this is important, origin and evolution of metabolic pathways, and gene duplication is not an explanation. When you start looking at it and seeing what's going on, it is not an explanation at all. Alibi is an evolutionist. Swift, I like what Dennis Swift said, even though this is a long time ago, it's still in the 21st century. So let me just cut to the chase and then we'll open up for questions. What is not said is that until the duplication acquires a new useful function, you've got this one gene here, and then sometimes the genes can duplicate. Well, so what? Now you have 2 of the same genes, okay? That doesn't give you more information. That doesn't give you more information. I can say that 5 or 6 times. That doesn't give you more information, okay? It just means that the gene is duplicated. There is no new additional information. So Swift is very good. I've had email contact with him. It confers little or no selective advantage because a simple duplication offers nothing new. So gene duplication, take it off the list. It's not an explanation. Well, let's see. Yeah, this comes from the Journal of Creation. Sometimes I'll quote creationists because they make a very good point. This is Leninger again, gene duplication and mutation, one path to generate new enzymatic activities. Gene duplication doesn't give you new enzymatic activities. You just have a duplicate gene, period. It doesn't give you anything new, but Leninger says it does. So let me cut to the chase and then we'll open up for questions when we get the lights on here. I just want to remind you what it says in Psalm 139, where the psalmist says in verse 14, I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, such as you see in this incredible biochemical display here. You just want to drop to your knees and say, hallelujah, hallelujah, how great thou art. Okay, thank you, Michael.